people still dispute the origin of life through natural processes. This is something I'm sometimes amazed at, but other times I can be a little more forgiving. Due to our anthropocentric bias, design is the way we conceive of the origin of, well, anything. Our cognition was productively honed by seeking and understanding agency in our environment, be it from predators, prey, or even other humans. But the very fact of our existence is due to natural selection and self-organization, processes that aren't explicable using a model of agency. These are things that just happen. So to people who can't step past this anthropocentric bias and build a naturalistic framework, what proof do we have to offer them to counter this intuition? You can't exactly put out a lawn chair on a perfectly tuned prebiotic world and just wait a billion years. And we can all agree there isn't an evolve faster button, right?
Are you saying Lieutenant Paris is evolving?
The only difference between natural evolution and what happened to Mr. Paris is that his changes took place over a 24-hour period. Traveling at infinite velocity accelerated the natural human evolutionary process by millions of years.
And what do we do when we get upset at bad Star Trek episodes?
Derail my entire video.
No.
So without our evolve faster button, what are we left with? How can we demonstrate evolutionary principles? Well, today I'm going to provide an interesting example of molecular evolution that scientists around the world can perform in a matter of months. Or a couple weeks if you have sufficiently motivated grad students. So we're going to talk about a method developed by researchers that utilizes evolutionary principles, serving as an analogy for natural selection. The method is called SELEX, aka the systematic evolution of ligands by exponential enrichment. Scientists and our acronyms, am I right? Now SELEX isn't just a proof of principle. Its main intention is to discover new molecules for both therapeutic and research purposes. Molecules we call ligands. Check our acronym, that's why it says the evolution of ligands. So what's a ligand? A ligand is just a type of molecule that binds to another molecule. It's just like that. And in a biochemical context, it means a smaller molecule that binds to a larger protein. You may not know it, but you've probably heard of ligands before. Think most of the active ingredients for drugs you're familiar with. Ibuprofen, penicillin, THC, all of these are molecules that bind to proteins and, by binding, influence their function, creating interesting downstream effects. So when people use SELEX, they're trying to find new ligands, stuff that sticks to proteins that we want to impact. Some examples of proteins we would want to target are KRAS, a human receptor involved in the development of cancer, or the ACE2 receptor, which mediates the entry of coronavirus into our cells. So by finding a ligand, you're finding a molecule that can be used as a drug to manipulate human physiology through the proteins they stick to. Now, this is a pretty typical drug design goal, but what we want to talk about today is the methodology. That's the interesting part. When SELEX searches for ligands, it doesn't search for small molecules like ibuprofen or penicillin. It uses single-stranded nucleic acids like RNA or DNA. You know, the stuff that carries information in our bodies using sequences of nitrogenous bases. You see, nucleic acids aren't just information carriers. They're molecules that have shapes and functions, and they can bind stuff too. When we think about the central molecules of life, we tend to give proteins the functional role, while nucleic acids get treated like a hard drive. But it's important to note that nucleic acids have the potential for direct functionality, as shown by several physiological examples. So if you had 10 random RNA molecules with different sequences of nucleotides, usually represented by the letters A, C, G, and U, they would all have different 3D conformations, different shapes, each defining a unique functionality. They basically would ball up, like headphones, folding in on themselves to form a conformation determined by their sequence of bases. And this is exactly what SELEX takes advantage of. Nucleic acids have information that define their function, and they can be directly replicated, unlike other biopolymers such as protein. So that's the background. But what does SELEX do exactly? Well, first, SELEX starts with a large library of different RNA sequences. But how large is large? Well, the average starting library actually has 10 to the power of 15 unique sequences, a quadrillion.
That's 1,15 zeros.
That's the number of grains of sand it would take to fill the Sydney Opera House, or the accumulated wealth of 5,700 Jeff Bezoses. Jeff Bezos? Now, given how large 10 to the 15 is, the assumption is that one of the sequences in this vast library has the right shape to perfectly bind to our target protein, and we call this special sequence an aptamer, the Cinderella for our protein glass slipper, so to speak. So how do we find this needle in a haystack? Well, if the goal is finding the stickiest sequence, why not start with throwing this library at our target protein and seeing what sticks? And this is what researchers do. They incubate the starting library with our target protein, and some RNA sequences bind and some don't. Researchers then wash away all the sequences that didn't stick and keep the ones that did. And voila! Our first round of selection is done, and we've already winnowed our pool down to sequences that bind to our target protein. But we're not done yet. We can select for even tighter binding using competitive iterative selection in a way that's directly analogous to natural selection. As I mentioned earlier, nucleic acids can be replicated. In the lab, we do this using a technique called PCR, the polymerase chain reaction. It's not super important to understand how PCR works, to understand the basic idea of SELEX. But just know that PCR is able to create perfect copies of a given DNA sequence, like a tiny molecular copy machine. So after we get our first set of sticky sequences, we amplify them using PCR, building our library back up. And now we're ready for round 2. This is the exponential enrichment portion of the acronym. We create more of what worked, and then you throw that new pool back at the target protein. See what binds best, rinse off the rest, etc., etc. SELEX is an iterative process of selection, enrichment, and then selection again, each time getting a stickier, more tightly binding set of sequences. And eventually, after 15 or so cycles of competition, a few sets of sequences start to dominate, and you can then test them individually to find that tightly binding ligand that fits less like Velcro and more like a key in a lock, our very own aptamer. It's less survival of the fittest and more survival of the stickiest. Now, hopefully, y'all already see the analogy to natural selection. A cycle of SELEX is like a generation of organisms being born, reproducing, and dying. Like, we could have replaced the RNA molecules in our diagrams with pictures of bacteria or bunnies, and the idea would be the same. Let's say you have a population of rabbits with a diverse set of traits, as shown here by using different colors. Now some selective pressure comes along that biases towards certain members of that population. Those members of the population then go on to create more offspring than the rest of the bunnies. If these conditions stayed the same for generations, you would end up with a population of rabbits that was highly adapted to the selective pressures of their environment.
Let's see it purple! Come on!
In SELEX, the selective pressure was being tightly binding, being sticky. Removal from the gene pool was accomplished through washing away the rejected sequences, and replication was, well, replication, just by PCR and not by biological reproduction. And this is why I consider the SELEX method to be a pseudo-example of evolution. It's a microcosm of a process that usually spans millennia. And it's also a fantastic example of how the concept of evolution can be applied in different contexts to produce interesting or useful results, just like genetic algorithms, or Twitch Plays Pokemon. Now maybe people won't see SELEX as direct evidence for evolution, and I would agree with you there. It's a good analogy, and an illustration of evolutionary principles. But actually, researchers have used the SELEX method to make a direct connection to the early evolution of life on Earth, or at least a direct connection to the incumbent hypothesis for early evolution, the RNA world hypothesis. In the RNA world hypothesis, the earliest organisms were actually RNA molecules which were able to replicate themselves. So they were both the information that was carried to the next generation and the means by which that information was replicated. They carried information that defined their function. Should sound familiar. Researchers at UCSD thought so too, and used a modified version of the SELEX protocol that produced RNA molecules capable of performing a requisite step in their own self-replication. Specifically, they found RNA molecules capable of catalyzing the addition of triphosphate to the 5' RNA hydroxyl group from cyclic trimetaphosphate, which would be a precursor step to self-polymerization. But we can discuss this more in office hours. This process of molecular evolution, performed in a laboratory, is likely not what happened as the first self-replicating molecules developed billions of years ago, but it's pretty much the closest thing we have to historical prebiotic RNA evolution. And that's the coolest thing about SELEX and in vitro selection in general. A version of it may have, potentially, sort of, happened billions of years ago. And by adhering to methods that have fidelity to the actual history of life, Moretti and Müller have given themselves the best possible chance of discovering authentic, self-replicating RNA molecules that are, in essence, our ancestors. And that's what an elegant and beautifully evocative experiment looks like. Let's remember our dichotomy from earlier, design versus self-organization. In terms of the evolution of life, this dichotomy is valid, and the evidence points firmly at natural selection. But the way we discover things, new inventions, new ideas, requires our agency, our design. However, the SELEX method, a method of discovery, bridges the design-self-organization dichotomy. It is a method designed to self-organize by hijacking the principles of evolution. Now, one could argue that this is nothing new. Consider the thousands of years of artificial selection that domesticated plants and animals have been subjected to. But that wasn't deliberate in the same way that SELEX is, which makes one wonder, how much can we manipulate our environment now that we better understand biological principles? Honestly, the possibilities are both amazing and potentially horrifying, but let's leave that for another day. Thanks for watching, and as always, please like and subscribe. Tornado. Run, Toto, run!